Hello again. Here we are in chapter 20, dealing with DNA tools and biotechnology. So we're going to be talking about some specific techniques in what we call the realm of biotechnology. So what does biotechnology actually mean? In a brief way, biotechnology is where we utilize our understanding of biological systems, particularly at the molecular level, to study or generate new products and materials. So we study products, study genes in order to identify genes that may be desirable for things like agriculture or medicine, study genes in order to understand ancestry or to identify someone, like in the case of forensics or somewhere. And so we're going to use the systems that we've studied a little bit in cell biology here in order to then apply and do work in society. Now, chapter 20 is much bigger and much more comprehensive than we can cover in a 100 level cell biology class. So we're going to narrow it down to four things. And so you will only be responsible for the reading in chapter 20 on these four things. The four things are PCR, which stands for polymerase chain reaction, where we can purify and amplify specific DNA sequences. Restriction enzymes, a key aspect of technology allowing us to very selectively cut DNA in very specific locations. Gel electrophoresis. It turns out that most biomolecules have a slightly negative charge. They are mild organic acids, and when they give up that proton, they have a slight negative charge, and so they will migrate in an electric field. And so we use a gel in order to basically create a sifting system, if you will, that can sort biomolecules by size, since they run to the negative charge, and if they're smaller or more compact, they will migrate faster. So it's a wonderful way to isolate, identify, and purify biomolecules, nucleic acids in particular, as well as proteins and other molecules. And then the final technology is called the CRISPR-Cas9. It's the most recent and arguably significant advance in biotechnology in the last 10 or 15 years. Before we get started into the biotechnology, though, you have to define a few things. By the way, this video here that you see, or this box you see here, is actually an interactive video that you can access in the PowerPoint. So please download the PowerPoint and watch these videos. You will be responsible for that material. And so this technology here, this definition, recombinant DNA, is the idea that we can recombine DNA. We can add, subtract sequences to other pieces of DNA. Along the lines of recombinant DNA is the idea of the what are called plasmids. Plasmids we find mostly in bacteria, but we do see them in eukaryotic cells as well, and we can actually exploit that. The idea here is that and again, this is back to recombinant DNA, is that bacterial DNA will occasionally split out and form these plasmids, these small donuts of DNA. We might have hundreds of thousands of nucleotides in the main bacterial chromosome, and we may only have, say, 10,000 base pairs or 5,000 base pairs in the plasmids. This is a way that bacteria can share genes across, not only across different strains of bacteria, but also even different species in some cases. This is sort of almost like a very, very early form of sort of sexual reproduction, if you will. We can see antibiotic resistance spread across species, and it's primarily the gene that confers that resistance gets put into one of these plasmids and gets shared to other bacteria. So we can and they can recombine, so these plasmids can then rejoin the main chromosome. But this idea here meant that by understanding that, we could then use it for our purposes of manipulation. And one of the things we've developed is we've developed these artificial donuts of DNA that behave like plasmids and can be introduced into bacteria in order to introduce new DNA to that bacteria. These artificial plasmids are known as cloning vectors. Cloning is making a duplicate copy. If you remember, if we create a clone organism, it's an exact genetic copy. We can do gene cloning, where we can insert a gene into one of these artificial plasmids and begin using it as shall we say, a gene Xerox machine, if you will. Bacteria become our DNA Xerox machines. And so here's a little overview of how this works. We identify a sequence of interest, and we clone those sequences using something like PCR or some other method, and then we purify that piece of DNA. We then, using ligase, if you remember ligase from DNA replication, and we'll talk about restriction enzymes a little later, we will then create a new donut of DNA with that piece of DNA inside. It's called a recombinant DNA plasmid. So we've created a donut of DNA. Now that plasmid will also have a gene in it that will allow us to select for a cell that has been what we call transformed. When we introduce DNA into the cells, not every cell will get that DNA. So this cell over here is not transformed, and what we have is we have something inside this that will then allow us to identify those that have been transformed and those which haven't. The most common way that we do this is to introduce an antibiotic resistance gene, like to ampicillin. So if we introduce the DNA to the bacteria, only the cells that receive the plasmid will be resistant to ampicillin, and the ones that don't have the plasmid will be killed off. And so we select for the cells that have been quote unquote transformed. And this recombinant technology has widespread applicability, not only in bacteria, but also in eukaryotic cells. Now we use bacterial plasmids. Why? Because they're easy to get. They're very easy to manipulate. You can make them quite quickly, and they're easy to purify. And they're easy to introduce. So we can actually, let's say we want to amplify a gene, or we want to do something along those lines, we can then grow them up in bacteria and make just boatloads of plasmid DNA for research or medical or other purposes. For instance, you can create a plasmid that has the DNA sequence. That means that if you put it into a eukaryotic organism, it will then cause the eukaryotic cell to then make a protein for us. That's why it can be used for medical purposes and that sort of a thing. I'm showing you here where we have plasmids, and then I'm showing you where even plasmids themselves can recombine into the original chromosome, and this is what we call a stable transfection. Now these cells will continue to hold on to that DNA. Whereas some cells that only have the plasmid will sometimes kick those things out. That's a little bit of an aside, but that's why I included this figure here. So let's add a piece to the puzzle now. We said that we could introduce DNA into a bacterial cloning vector. Okay. Well, how do we do that? Well, we have something that we discovered probably about, it's been about 40 years or so. It's been a little while. We found that bacteria have defenses against viral infection, and how it works on a virus is that they have enzymes that will only cut viral DNA sequences. They won't cut bacterial DNA sequences. They'll only cut viral DNA sequences, and so they call them restricted to viruses or restriction enzymes. These enzymes will cut DNA, and they will cut it only at very, very specific sequence sites. Like, for instance, this restriction enzyme is one of the original ones, E. coli. ECO restriction 1, ECOR1, will cut this sequence right here, this GAATTC. but it won't cut it in a square cut. It cuts it in an overlapping tongue, if you will, like a tongue and groove kind of a setup. These ends will want to recombine. Why? Because they have complementary DNA sequence. So they will occasionally recombine on their own, but we call that desire to sort of that staggered cut. We call those sticky ends. Now we have restriction enzymes that will do blunt end cuts, and those can be used as well. It's just they're not as efficient. These EcoR1 sites and these sort of staggered end cuts make for very, very easy and selective insertion. And here's just another zoomed in picture. That sticky end, the reason they call it sticky is because it does have a propensity to recombine with another sequence of this kind. Now what you can do is you can create an artificial sequence that then has those ends in there and will occasionally fit right into this, and so you can insert DNA in between these two sticky ends. That's how we add the additional DNA into a cloning DNA vector. So how this works is that you have your vector of choice. This one happens to be a vector that will confer a resistance to ampicillin and also neomycin. So it's got a double resistance vector here. We've got the promoter. If you remember, a promoter is the DNA sequence that leads to transcription. And this one's a promoter from a virus, a cytomegalovirus. It's a very, very strong promoter. It will really drive the cells to transcribe whatever is downstream from it. So, and then we have this area here where we can insert. We have several sites. This JO1 and EcoR1, HND3, and NOD1. Those are all different restriction enzymes. So you can pick one of these restrictions, or you can pick a couple of them if you want, and you will cut the DNA right there, and then you can insert that piece of DNA into that spot. So we digest in this case, we're going to use, we have an EcoR1 here and a NOT1 site. That means that we are only going to get an orientation this way. We're not going to get the inverted orientation. If you use the same on both sides, you could get backwards insertion, and you have to be careful about that. So we created a little DNA sequence here that has EcoR1 sticky end on one site and a NOT1 sticky end on the other site. We chop out that little piece of DNA right there at the EcoR1 and NOT1 site. Then we use DNA ligase to bind to fix the DNA, and some of these vectors may spontaneously reanneal, but with EcoR1 NOT1 it's not likely to happen. They probably won't recombine on their own. The only ones that will recombine in the vast majority of cases will be the ones that have the insertion in here. But you're going to double check that with gel electrophoresis. So we've got this inserted. It's oriented in the way that we want it to. When we insert that new vector into bacteria, it will not only be resistant to ampicillin, but also resistant to neomycin. Here's another video for you found in the PowerPoint. Please do, again, download the PowerPoint. Don't watch it in Brightspace. Download it so that you can have access to these videos. They're very, very good and informative. So please take a few minutes to watch those videos. The really, really cool thing about DNA vectors, and this is part of the reason, this also feeds into the fact that the central dogma holds up. This actually reinforces the central dogma. The data that you can take DNA from almost any source, like for instance, no bacterium has aardvark DNA in it. No tomato has aardvark DNA in it. Well, if you have a plasmid and you cleave with a restriction enzyme, and you recombine this aardvark DNA sequence, you can artificially introduce aardvark DNA into another organism, maybe a bacteria or a corn plant or a tomato. That's this right here, this principle that you can take DNA from one organism and introduce it into another is the idea behind GMOs. GMOs are animals or mostly plants that contain DNA that is not of the original genome that's been inserted into that organism. And so there's a lot of misunderstanding about GMOs and a lot of misinformation out there. So hopefully we can alleviate some of your concerns, but also introduce some potential things that we need to be careful about with GMOs in some cases. So let's walk through the workflow here. We've got our bacterial plasmid. This is our vector. We have a restriction site that we've identified in the vector, and we pick the proper restriction enzyme to cut at that site. We get these sticky ends. We have used gel electrophoresis and PCR in order to create a small cloned sequence of DNA from maybe another bacteria or another organism altogether, like an aardvark. We then we combine those two molecules, the digested plasmid and the small oligonucleotide. We use DNA ligase to seal the strands, and we end up with a recombinant DNA molecule. We'll also purify this using gel electrophoresis, and then we will digest it again in order to ensure that we have the proper orientation and the proper insertion. I've been mentioning gel electrophoresis a little bit. We will talk about gel electrophoresis a little bit more in class. But remember that biomolecules generally have a slight negative charge due to their acidic nature, and they will move in an electric field. And so we use a gel. A gel is porous. It's like a filter. It's porous, and you can actually change the density of the gel in order to handle different sizes of molecules. In this case, we have a DNA gel that will allow us to sort DNA sequences as small as 200 base pairs and as large as 10,000. Now, what we do is we load the gel, and we'll show you this in class. There are lanes here where we can add DNA extraction along with a little bit of marker so that we can see the migration of the material. And we also put in what's called a DNA ladder. In the case of proteins, we use something similar. We use protein markers. And these ladders are a known length of DNA. So we use a known length of DNA. And the smaller molecules will move further. The larger molecules will move less. Because remember, size is one of the factors in diffusion. And we're just creating an artificial diffusion. We create an electric field, and the molecules will diffuse across that electric field through the gel. If it's a larger molecule, it'll run slower. If it's a smaller molecule, it'll run faster. By knowing how large or predicting how large the pieces are in our recombined plasmid, we can then identify whether or not that plasmid has the proper DNA in it, and also orientation as well. We can do some orientation experiments. So if it's uncut, it's about 4,100 base pairs, and we see it run somewhere around that range between 4,000 and 6,000 on our markers. When we cut it with our restriction enzymes, that piece will be cut out. And so we'll have a larger piece somewhere around 2,900 base pairs. And again, we see it running right around the 3,000 marker. And we have a smaller piece, about 1,200 base pairs. And again, we see that running between the 1,000 and 2,000. So we know we have an insert in there and that we don't have some other artifact. So here we have a picture of your typical gel electrophoresis. Here's the DNA ladder. Okay, we can see that. Here are the original wells that we load. We use a special pipetter to take the mixture of molecules of different lengths in our different experiments. We add those to the wells. Make sure you don't punch a hole in the wells when you do this. You add those to the wells, we turn on the power source, and the DNA will move towards the positive the positive anode because they're slightly negative. They will move towards the positive side of this. And so what we see are the different setups based on, like, for instance, this one looks like an uncut piece of DNA, and we've got a couple of plasmids that look like they have some inserts in them, you know, another one right here. And again, we have another molecular weight marker over here, so we can make sure that we have the right markers for the right the right size pieces. Gel electrophoresis. Now, I found a really good video from a company on restriction digest analysis, on how you use restriction enzymes and gel electrophoresis to identify if your plasmid has the insert and whether it's properly oriented. So here is the link here. I will be putting this link in bright space for you. Please watch this, because we will be doing something like this in lab, and I will be holding you a bit responsible for the material in this video. So we've just hit the 21-minute mark here, and I think that's long enough for a video. So this is the end of part one. What we will do in the next video is pick up where we leave off here, starting with polymerase chain reaction, a very powerful and important biotechnology tool. So thank you for your time, and hope you enjoyed the video so far, and I will talk to you soon.